Are you keeping up with the latest in spinal cord injury research? If not, you might be missing critical developments. Today, we're diving deep into NX210c, a peptide generating significant interest in the preclinical space. Spinal cord injury remains a profoundly challenging area, with current research strategies often limited to acute stabilization and rehabilitation rather than true neurological repair. We'll explore how NX210c offers a new, scientifically grounded approach in laboratory studies. Spinal cord injury remains a devastating neurological condition, affecting hundreds of thousands globally each year. Researchers actively seek pharmacological therapies to reliably restore motor function after such complex injuries. Most strategies currently focus on surgical intervention and long-term rehabilitation, not direct neurological repair. NX210c stands out, demonstrating multidimensional effects in animal models of spinal cord injury. This peptide improves motor function, tissue preservation, and neuronal signaling in preclinical studies. Derived from SCO-spondin, NX210c leverages thrombospondin-related biology to modulate the injury environment. SCO-spondin is a glycoprotein found in the central nervous system, expressed in the subcommissural organ. It contains thrombospondin type 1 repeat sequences, crucial for axon guidance and synapse formation. Thrombospondins are extracellular matrix components, vital for cell-to-cell and cell-to-matrix interactions. Their signaling is linked to axonal pathfinding and synaptic plasticity, essential for neural repair. NX210c acts as a bioactive peptide, modulating the post-injury environment in the spinal cord. It influences multiple aspects of secondary injury, including neuronal survival and tissue preservation. Spinal cord injury involves a complex cascade of secondary processes after initial trauma. Many experimental therapies fail by targeting only one aspect or requiring unrealistically narrow intervention windows. NX210c shows beneficial effects even when administered hours post-injury in lab settings. Earlier studies showed improved axonal regeneration in thoracic SCI models using this peptide. Recent work extends these findings to cervical spinal cord injury, a highly relevant research area. This multifaceted approach of NX210c truly highlights the evolving landscape of peptide research. It's crucial to stay informed about these advancements. If you're finding this deep dive valuable, remember you can follow me, Sonia Rao, for more specialized insights, and be sure to subscribe to Peptides Today for all general peptide news and pipeline updates. We're just scratching the surface of NX210c's potential. The most recent preclinical assessment of NX210c utilized a cervical spinal cord injury rat model. Animals received bilateral clip compression contusion injury at C6-C7, mimicking traumatic cervical SCI. NX210c was administered daily via intraperitoneal injections at 8 mg per kilogram. Treatment continued for 8 weeks, evaluating two clinically relevant initiation windows. One group started treatment 4 hours post-injury, another began at 8 hours. This design assessed both efficacy and the therapeutic window of the experimental peptide. Early NX210c administration led to significant improvements in forelimb grip strength in rats. Treated rats also showed improvements in static and dynamic locomotion, enhancing interlimb coordination. Delayed NX210c administration, 8 hours post-injury, yielded compelling findings. These animals exhibited improved weight gain, an indirect marker of systemic recovery and reduced stress. Trunk balance improved significantly, measured by the inclined plane test, crucial for stability. There was also a trend toward faster recovery of bladder control and skilled reaching performance. Histological evidence further supported NX210c's efficacy in the laboratory studies. Treated animals showed significantly greater white and gray matter preservation at the injury site. Cavity size at the lesion site was also reduced, limiting neural continuity disruption. Upregulation of neuronal markers in treated tissue supports neuroprotective or neurorestorative effects. The apparent benefit of delayed administration suggests NX210ca may act during the subacute phase. NX210c represents a scientifically credible and biologically rational approach for spinal cord injury research. Its ability to improve outcomes and preserve neural tissue positions it as a promising candidate. While transition from animal models to human research is challenging, NX210c's outlook remains strong. To keep abreast of these cutting-edge peptide discoveries, follow me, Sonia Rao, and subscribe to Peptides Today. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.